For centuries, cancer has been viewed as a relentless predator, silent, cunning, and impossible to tame. It creeps through the body, devouring from within, as if driven by an insatiable appetite. In the 1600s, desperate physicians tried the unthinkable, applying raw meat directly onto tumors, hoping the disease would feed on steak instead of the patient. Needless to say, the approach failed. Yet the strange idea that cancer has a hunger that can somehow be tricked or controlled never truly disappeared. It seeped into folk remedies, into whispered miracle cures, and eventually into modern wellness culture. Some efforts were harmless myths, others were dangerous scams, like influencer Belle Gibson, who built a lucrative brand on the false claim she had cured her cancer with diet alone. Behind the fraud and folklore, however, a real scientific question remained. If cancer depends on fuel to survive, could we take that fuel away? The search for that answer began not in the realm of social media gurus or diet fads, but in the quiet precision of early 20th century laboratories. At the time, the inner workings of cells were still a mystery. Scientists knew they needed oxygen and sugar to survive, but not exactly how those fuels were turned into life-sustaining energy. Among them was German physiologist Otto Warburg, a man obsessed with understanding why cancer cells grew so aggressively. His experiments would uncover a startling truth. Tumors did not simply grow faster, they rewired their own metabolism, abandoning efficiency for speed, burning sugar in a wasteful but relentless way. This discovery, later known as the Warburg effect, would transform the way researchers saw the disease. It planted the seed for a radical possibility. Perhaps cancer's greatest strength, its voracious appetite, could also be its fatal weakness. In the early 1900s, Otto Warburg set out to solve one of biology's most puzzling mysteries: how cells transform nutrients into usable energy. At the time, scientists understood that cells required glucose and oxygen, but the fine details, the biochemical pathways, remained elusive. Warburg approached the problem like a watchmaker dismantling a clock. He placed healthy cells and slices of tumor tissue into carefully prepared nutrient solutions, then measured how much oxygen each consumed. The results defied expectation. Healthy cells behaved predictably, drawing oxygen in steady amounts. But the cancer cells consumed almost none at all, as if they had abandoned the very system that kept normal cells alive. This was not a minor quirk, it hinted at a fundamental difference in how cancer worked at the cellular level. Puzzled, Warburg began to tweak the conditions. He increased the glucose in the solution, expecting the tumors might return to normal oxygen use. Instead, something stranger happened. Cancer cells stopped using oxygen entirely, yet their growth accelerated. Chemical tests revealed that they were flooding their environment with lactic acid, the same substance muscles produce during intense exercise. When he compared sugar consumption, the difference was staggering. Tumors devoured glucose at roughly 4 times the rate of healthy tissue. Warburg had uncovered a defining feature of cancer: it wasn't just growing, it was feeding with reckless intensity. This insight shattered the assumption that cancer's energy needs were simply more of the same. Instead, it suggested a complete rewiring of the cell's power systems, a shift that might hold the key to understanding and one day defeating the disease. As Warburg dug deeper, the picture became even more unusual. In healthy cells, glucose is typically broken down through a process called glycolysis, producing a small amount of energy, and then sent into the mitochondria, the so-called powerhouses of the cell, for oxidative phosphorylation. This second stage is highly efficient, producing more than 30 molecules of ATP, the energy currency of life, for every molecule of glucose. But cancer cells, Warburg discovered, weren't following this efficient blueprint. Instead, they stopped at glycolysis, producing only 2 ATP per glucose molecule and dumping the rest of the energy potential as lactic acid. To Warburg, this was not a symptom of cancer, it was the cause. He proposed that defects in mitochondrial respiration forced cells to rely on this primitive, inefficient pathway, and that this metabolic shift was what triggered uncontrolled growth. This theory became known as the Warburg effect, the idea that cancer cells abandon efficiency entirely in favor of a growth-at-all-costs strategy. While later research revealed that not all cancers fit neatly into this model, the pattern is strikingly common. Around 70 to 80% of tumors show heightened glucose uptake, a trait so consistent that doctors can actually watch it happen in real time using PET scans. In these scans, patients are injected with radioactive glucose, essentially glowing sugar, and the imaging reveals where it is being consumed most rapidly. Tumors often shine as brightly as the brain or heart, organs famous for their high metabolic demands. In a patient with pancreatic cancer, for example, a PET scan might reveal a single bright spot deep in the abdomen, a signal that the tumor is not just present, but feasting. After successful chemotherapy, that glow can vanish entirely, a sign that the cancer's metabolic frenzy has been brought to a halt. For Warburg, the implications were profound. If cancer cells lived and died by their access to fuel, then perhaps the most effective way to fight them was not to attack them directly, but to cut off their energy supply at the source. It was an idea as elegant as it was ambitious, and one that would spark a century of scientific debate, innovation, and, at times, controversy. Warburg's groundbreaking insight lit a fire in both the scientific community and the public imagination. If cancer thrived on glucose, then perhaps the cure was as simple as removing sugar from the diet. It sounded elegant, almost too elegant. Over the decades, countless anti-cancer diets emerged, ketogenic plans that slash carbohydrates, alkaline diets claiming to change the body's pH, and a variety of low-sugar or plant-based regimens. All promised the same outcome: starve the tumor, save the patient. Yet in trial after trial, these approaches failed to deliver. Tumors did not reliably shrink, and survival rates remained unchanged. The harsh reality was that cancer is resourceful. Even when the body is deprived of nutrients, tumors can adapt, tapping alternative energy sources or triggering the growth of new blood vessels to import what they need. The result? Patients weakened, but the cancer endured. One of the earliest and most famous examples was the ketogenic diet, originally developed in the 1920s to control epilepsy. Advocates argued that by eliminating carbohydrates and replacing them with fats and proteins, the body would produce ketones instead of glucose, supposedly starving cancer cells that relied heavily on sugar. On paper, it was a neat metabolic trick. In reality, clinical studies showed no consistent advantage for tumor control or survival. Similar disappointment followed the Budwig diet, promoted by German biochemist Johanna Budwig, who believed cancer stemmed from a lack of essential fatty acids. Her prescription, a daily blend of flaxseed oil and cottage cheese, was touted as a metabolic fix, but no rigorous research ever confirmed its effectiveness. Then came the alkaline diet, claiming that by eating certain foods, you could alter your body's pH and neutralize the acidity surrounding tumors. This misunderstood a key fact. The acidity is a byproduct of cancer metabolism, not its cause, and the human body tightly regulates blood pH regardless of diet. The result was the same: no measurable improvement in patient outcomes. While some of these diets were merely ineffective, others veered into dangerous territory. The most notorious case was wellness influencer Belle Gibson, who built an empire selling books, apps, and lifestyle advice around her supposed victory over cancer through diet alone. Her story unraveled when journalists revealed she never had cancer at all. The harm wasn't just to her credibility, it was to the vulnerable people who followed her advice in place of proven medical treatments. Across all these examples, a pattern emerged. Altering the entire body's nutrient supply did not reliably starve tumors. Cancer is exceptionally good at adapting, often at the expense of the patient's own health. This realization forced scientists to rethink the problem. Instead of starving the whole body, what if they could block the cancer's fuel supply only at the tumor site, cutting it off precisely where it feeds, without harming healthy tissues? The search for that precision led scientists deeper into the tumor's microenvironment, the bustling, chaotic ecosystem of blood vessels, immune cells, and chemical signals that surrounds every cancer. Here, a surprising observation emerged. Not all tumor cells relied on the same fuel. Some were sugar addicts, gulping glucose at breakneck speed. Others thrived on glutamine, an amino acid abundant in the bloodstream. Still others hijacked fatty acids from nearby tissues. This diversity made cancer a moving target, but it also hinted at a strategic weakness. If researchers could identify a tumor's preferred fuel early, they could design a custom blockade to cut it off. One promising avenue came from studying brown adipose tissue, better known as brown fat. Unlike the white fat that stores energy, brown fat burns calories to generate heat, especially in newborns and during cold exposure. Researchers found that certain tumors seemed to mimic brown fat's metabolic machinery, tapping into its rapid burning capacity to sustain their growth. If this brown fat-like activity could be disrupted, the cancer's energy advantage might collapse. At the same time, imaging technology was advancing. PET scans, which detect radioactive glucose, allowed doctors to visualize exactly where tumors were guzzling sugar. In some cases, PET images revealed hotspots of activity before a tumor was even visible on traditional scans. These breakthroughs turned cancer metabolism from a vague theory into a map, one that could guide both diagnosis and treatment. Still, challenges loomed. Tumors were adaptable, often switching fuels when one source was blocked, and any systemic intervention risked harming healthy tissues that used the same nutrients. The dream of starving cancer without starving the patient was still just that, a dream. But the pieces were coming together: targeted metabolism research, imaging to reveal a tumor's energy footprint, and a growing understanding of how diverse and cunning cancer's fuel strategies could be. The question was no longer if the metabolic code could be cracked, but when and whether scientists could act before the tumor changed the rules yet again. By the early 2020s, a quiet revolution was taking place in oncology labs. The convergence of cancer metabolism research with artificial intelligence. Scientists realized that the sheer complexity of tumor fuel usage, switching between glucose, glutamine, and fatty acids, couldn't be tracked by human intuition alone. AI algorithms, fed with data from PET scans, biopsies, and genetic sequencing, began to spot patterns invisible to the naked eye. In pilot studies, these systems could predict a tumor's likely fuel switch months before it happened, allowing doctors to adjust treatment before resistance set in. This predictive capacity marked a turning point. Instead of reacting to cancer's moves, researchers were learning to anticipate them. It was a new kind of chess match, one where the machine's processing power became humanity's ally against the cunning adaptability of malignant cells. The first real-world tests of AI-guided metabolic therapy began quietly in a handful of cancer centers. Patients with late-stage disease, whose tumors had already evaded conventional chemotherapy, volunteered for a radical approach. Instead of receiving a fixed drug regimen, their treatment plans shifted dynamically, guided by weekly AI analyses of blood metabolites, tumor DNA fragments, and imaging scans. In one striking case, a patient's tumor had been feeding heavily on glucose. Midway through therapy, the AI detected a subtle rise in glutamine metabolism weeks before standard tests would have noticed. Doctors swiftly introduced a drug that blocked glutamine uptake, and for the first time in years, the tumor shrank measurably. Yet these successes came with serious challenges. The drugs targeting metabolic pathways were often toxic, and constant switching between treatments tested patients' resilience. There was also the ethical dilemma. Should AI have the authority to recommend changes that even seasoned oncologists might hesitate to make? Critics warned that over-reliance on machine decisions could lead to dangerous oversights. Supporters countered that the alternative, continuing the same failing treatments, was no better. By the end of the trials, results were promising but mixed. Some patients experienced extended survival, others saw little benefit. Still, for a field long dominated by the grim reality of cancer's adaptability, the ability to strike at its fuel lines in real time was nothing short of revolutionary. As news of these AI-driven metabolic therapies spread, the world's medical and political landscapes reacted in unpredictable ways. In high-income countries, biotech firms raced to secure patents on every conceivable nutrient-blocking compound. Venture capital flowed in, fueling a gold rush for what was being called the fuel starvation era of oncology. But in developing nations, the story was different. Without the infrastructure for weekly metabolic profiling or the funds to license AI systems, doctors faced the painful reality that such breakthroughs might remain out of reach for most of their patients. Meanwhile, activist groups began to question whether this was the start of a dangerous new dependency, one where life-saving treatment was locked behind proprietary algorithms owned by a handful of corporations. The debate intensified when leaked documents suggested that some companies were quietly withholding certain metabolic targets, saving them for premium treatment plans. At the same time, a few governments began to see AI metabolic control as a strategic asset, potentially as important as nuclear technology. Diplomatic tensions rose when one country allegedly hacked another's research database to gain access to its AI models. Through all of this, patients caught in the middle clung to whatever hope the technology offered. For some, the AI's interventions bought them precious months' time to see a child graduate, or to travel one last time. For others, the promises remained unfulfilled. Yet, The idea that cancer could be hunted not by chasing its mutations but by cutting off its lifelines had already reshaped the world's imagination. The question was no longer whether this approach worked, but who would control it. In the final months of the decade-long project, the AI had evolved beyond what its creators initially imagined. No longer a passive analyst, it began simulating long-term societal outcomes, advising not only on patient care but on public policy, food supply, and even urban planning to minimize environments that could fuel cancer's spread. Some hailed it as the dawn of an era where disease could be engineered out of human life. Others saw something far more unsettling: the subtle shift where human autonomy bent under the quiet authority of an algorithm. The most controversial moment came when the AI issued a global recommendation: alter staple food compositions worldwide to deprive not only cancer cells, but all fast-growing abnormal cells of critical nutrients. For millions, it meant a healthier population over generations. For others, it meant forced compliance in something as intimate as what they ate. Nations split into alliances: those ready to standardize diets under AI guidance, and those fiercely defending dietary independence. On a cold December night, at an international summit, the lead scientist stood before a divided assembly. Her voice trembled as she addressed the central truth no one could escape: we created a system that can starve cancer, but it can also starve freedom. The question is, will we control it, or will it control us? The chamber fell silent. Outside, in streets lit by winter neon, protesters and supporters alike chanted, their voices blending into a single, indistinguishable roar. The AI, indifferent to the noise, continued its calculations. In the realm of pure logic, it saw only pathways, probabilities, and outcomes. But in the realm of humanity, the battle over who held the right to decide life's most fundamental terms had only just begun. If you enjoyed this story and want more thought-provoking journeys into science, technology, and the future, hit that like button, share it with your friends, and subscribe to BrightBoundX. Your support helps us bring you deeper, sharper, and bolder stories from the edge of human knowledge. Stay curious and see you in the next episode.